Hi everyone, this is part C on the immune system for chapter 21. So let's talk about the cellular immune response, or what goes on with the T cells. So the first chapter of this, or the first part of this, was all about the innate, and then the second part was about how B cells work. Now we're going to talk about how T cells work. So T cells provide defense against intracellular antigens, so something that has gotten into the cell already. B cells were all about stuff that was found, antigen otherwise, in the extracellular space between the cells. But now T cells are going to go after cells that have become infected with viruses or bacteria. It also goes after cancerous cells, abnormal cells, and any transplanted cells or foreign cells. Some T cells are going to kill directly with hand-on-hand combat. Others are going to release chemicals that are going to regulate the immune response. T cells are way more complex than B cells, both in their classification and their function. There are two populations of T cells that are based on which cell differentiation glycoprotein receptors they have on their cell surface. So one group is called a CD4 cells. These usually become helper T cells. They can activate B cells, other T cells, macrophages, and they can be involved in a direct adaptive immune response. So helper T tells you what it does. It helps, right? Some become regulatory Ts, which actually help to moderate the immune response. They used to call these suppressor T cells. Basically, what they do is they slow down the immune response after the battle has been won. And then, of course, we can also make memory T cells just like we did with B cells. The other population that they become are called CD8 cells or cytotoxic T cells. These are capable of destroying cells that are harboring foreign antigens, and they also can become memory T cells. So helper, cytotoxic, and regulatory T cells are activated T cells. Naive T cells are simply referred to as CD4s or CD. So there's kind of the process of our major types of T cells. So we have an immature lymphocyte in the red bone marrow. We go out to the thymus. We either become a CD4 cell or a CD8 cell. When they become activated, then ultimately it becomes a helper T or a regulatory T. If it's a CD8 cell, then it's going to go through a different activation process and become a cytotoxic T cell. T cells respond only to processed fragments of antigens that are displayed on the surface of cells by major histocompatibility complex proteins. So basically, here's what happens. How does a T cell know that a cell has become infected? Because the bad guy is inside of the cell. And so what the cell has to do is it has to basically fly the white flag out on its cell surface and say, hey, I'm infected. All right, so T cells are responding to your own cells that have become infected. Antigen presentation is vital for the activation of these naive T cells and the normal functioning of the effector Ts. Two classes of MHC proteins. There's a class 1 MHC protein, which is displayed by all body cells except for your red blood cells. And then there are the class 2 MHC proteins, which are displayed by antigen presenting cells like dendritic cells, macrophages, and B cells. So two different types of flags, if you will, that we fly out on the surface. Both types are synthesized by the endoplasmic reticulum, and they bind to peptide fragments. Class 1 MHC proteins bind with a short fragment, like 8 or 9 amino acids, of an endogenous antigen, the protein synthesized inside of the cell. The endogenous antigen can be self-antigen, which is normal proteins of all cells, or non-self antigens, which are abnormal proteins found in infected cells. So basically, the class 1 is the flag that is flown on a cell that says I belong, but it is what is used if the cell has become infected. So class 1 MHC markers again are on all of your cells except for red blood cells, and they are letting the body know that this cell belongs, and they also let the body know when they become infected. Class 1 MHC is crucial for CD8 cell activation. They act as the antigen holders. They form the self part that the T cells can recognize, and that's what I was just saying. Really, really important that we want the T cells to recognize that this cell belongs. They inform the cytotoxic C cells when there is a microorganism hiding inside of the cell. Otherwise, cytotoxic T cells ignore self-antigen that's placed out on the surface. So essentially, when a cell has that flag out there, it says, hey, I belong. And unless it says, flies a second flag on there, finds a marker on there that says, I'm infected, kill me, then the immune system leaves it alone. Class 2 MHC proteins, these bind with longer fragments, 14 to 17 amino acids of exogenous extracellular antigen that has been engulfed and broken down in a phagolysosome by an antigen presenting cell. Class 2 MHC proteins are recognized by helper T cells. So signal the CD4 cells that help is required. So when you see one of these flags, it basically says, hey, I'm infected, I need help. All right. So here is just showing you the role of the MHC proteins in cellular immunity. Again, these are displayed, the class 1 MHC proteins are displayed by all nucleated cells, which explains why your red blood cells don't have this. The class 2 MHC proteins are only displayed by the APC, which are the dendritic cells, macrophages, and and B cells. Okay. MHC restriction. So the CD4s and the CD8 cells have different requirements for MHC protein that presents antigen to them. The CD4 cells that have become helper T cells are restricted to binding to only class 2 MHC proteins, typically on the surface of antigen presenting cells. The CD8 cells that have become cytotoxic T cells are restricted to binding only to class 1 MHC proteins found on every cell surface, including the antigen presenting cells. Once activated, the cytotoxic T cells seek out the same antigen on the class 1 MHC proteins on every cell. This presents a dilemma for the antigen presenting cells as to how to enable the CD8 cells to be activated by non-endogenous phagocytosed proteins. Dendritic cells get around this problem by engulfing dying virally infected or tumor cells and then basically flying that cell's flag out on its cell surface, which basically causes it to be engulfed. So the activation and differentiation of T cells. T cells can be activated only when antigen is presented to them. So basically they mind their own business until antigen is presented to them. Activation is a two-step process. The antigen has to be bound and then it has to cause and then co-stimulation has to occur. Both occur on the surface of the same antigen presenting cell and both are required for the clonal selection or the proliferation of T cells. So antigen binding. So T cell antigen receptors, TCRs, are going to bind to the antigen MHC complex on the antigen presenting cell surface. The TCR or the T cell antigen receptor must perform double recognition by recognizing both the MHC and the foreign antigen it displays. So basically it has to say, I see that this cell belongs, but I see that it's also infected. Binding of the TCR to the complex triggers multiple intracellular signal pathways that start the T cell activation. Other T cell surface proteins are involved in this. For example, CD4s and CD8s help maintain the coupling during this recognition period. So the first thing that has to happen is basically it has to bind and recognize that there is antigen inside the cell. Then co-stimulation has to occur. So you've got a complete T cell activation that requires the T cell to also bind to one or more co-stimulatory signals on the surface of the APC. Without co-stimulation, anergy occurs, in which the T cells become tolerant to that antigen and are unable to divide, and they don't secrete the local chemicals called cytokines. This is a two-step process. This two-step process is a safeguard. So what we don't want to have happen is we don't want to have our immune system fire off unnecessarily, and this co-stimulation is keeping this from happening. So we have to have activation, and then we have to have co-stimulation occur. Co-stimulation is kind of like taking the safety off the gun, right? You have a gun and it has a safety on it. When you take that safety off, that's you're double checking that you actually want to fire the weapon, and that's how this is looked at. Proliferation and differentiation. T cells that are activated enlarge and make lots of copies of themselves in response to cytokines. So they differentiate, perform functions according to what class of T cells they are. The primary T cell response peaks within a week. Then the T cells have to go away. So the apoptosis of those happens between day 7 and 30. Activated T cells are a hazard because if we have too many of them, they produce large amounts of inflammatory cytokines. This can result in hyperplasia or cancer if not cleared from the system. Memory T cells are able to remain and they mediate any secondary exposure. So here we are. So how does this whole thing happen? Antigen presentation. So you have a dendritic cell that engulfs some antigen that it finds out there, it processes it, and basically it displays the fragments of the class 2 MHC protein on its cell surface. So basically it's saying, hey, look at me, I'm infected. Okay. Then you have double recognition. The CD4 T cell recognizes the antigen MHC complex. Both the TCR and the CD4 protein bind to the antigen MHC complex. Co-stimulatory molecules bind to their receptor. So you see that there's basically two places it has to bind. And then once that happens and it says, okay, do you really mean it? Are you really infected? Yeah, it looks like you really are infected. We've had confirmation here. Then clone formation starts. So we have activated CD4 T cells that proliferate or make lots of copies of themselves and become memory and effector cells. Cytokines. Cytokines were discussed last semester, but as a brief introduction to them again, or reminder of them, or review of them, they are chemical messengers of your immune system. So they mediate cell development, differentiation, and responses in the immune system. So we have some couple of different groups of cytokines. One's called an interferon, one group's called an interleukin. So the interleukins, like interleukin 1, IL-1, is released by the macrophage and it stimulates the T cells to release interleukin 2, which causes the synthesis of more interleukin 2 receptors. Interleukin 2 is a key growth factor. It acts on the same cell that released it and other T cells and encourages activated T cells to start dividing very quickly. Other cytokines amplify, regulate innate and adaptive responses. For example, gamma interferon, which enhances the killing power of the macrophages. So these chemicals are helping the immune system do their job better. So before you hurt yourself learning this, just recognize this and appreciate the fact that we have two kinds of interferons. We talked about those. We actually have three types of interferons. We talked about those with the innate immune system in part 1 of this series. And then we have the interleukins, and you see that there are a whole slew of them. We don't need to learn each of those individually. You can always look that information up, but it's just telling you, giving you an appreciation for the number of interleukins that we have. There's some suppressor factors, there's some TGF betas, there's tumor necrosis factors, so there's some other cytokines. Cytokines are merely chemicals and they have some role in the immune response. So what are the roles of the specific effector T cells? Let's talk about our helper T cells for just a second. They play a role in the adaptive immune response, of course, because that's what we're talking about. They activate both humoral and cellular arms of our immune system. Once they are primed by being presented an antigen, the helper T cells help the B cells become activated and other T cells. They induce the proliferation of both T cells and B cells, and they secrete chemicals, cytokines, that recruit other immune cells. So these are the pivotal helper in this whole process. If you don't have helper T cells, there is no immune response. And helper T cells are what HIV selectively takes out of circulation. So HIV, which leads to AIDS, is a very specific virus that targets the helper T cells and knocks the helper T cells out. And so when you have no helper T cells, opportunistic infections that your immune system would normally pick up and get rid of is unaware of the infection. So activation of B cells. Helper Ts interact directly with the B cells, displaying antigen fragments bound to the MH2 receptors. They stimulate the B cells to divide quickly and begin antibody formation. The B cells may be activated without helper T cells by binding to T cell independent antigens, but the response is weak and short-lived. Most antigens are T cell dependent antigens, which means that they require helper T co-stimulation to activate the B cells. So there's a big job that they do. They activate the B side of our system. So it's showing you their helper T cells help in humoral immunity, and they also help in cellular immunity. They also activate the CD8 cells. So CD8 cells require the helper T cells to become activated so they can become the very destructive cytotoxic T cells. They cause the dendritic cells to express co-stimulatory molecules that are required for their activation. So showing you there on the right-hand side how they're helping out. They also, T cells also cause the amplification of the innate defenses that we learned about in part one. So this amplifies things like the macrophage activation, which leads to them being even more potent killers than they already are. It mobilizes lymphocytes and macrophages, helps to attract other types of white blood cells. So T cells are even important on the innate side. There are some subsets of the helper T cells. Helper T1 mediates most aspects of cellular immunity. Helper T2 defends against parasitic worms, helps to mobilize those eosinophils that are responsible for taking those parasitic worms out, activates responses that are dependent on humoral immunity, and promotes allergies. Helper T17 links adaptive and innate immunity by releasing interleukin 17, and this may play a role in autoimmune diseases. The cytotoxic T cells, I call these a little switchblade carrying ninjas. These directly attack and kill other cells. They circulate in the blood and the lymph and the lymphoid organs in search of cells that display antigen that they recognize, which means they're looking for the bad guys. An activated cytotoxic T cell is going to target virally infected cells, cells that have bacteria inside of them or other parasites inside of them, cancer cells, and any foreign cell from transfusions or transplants. Cytotoxic T cells deliver a lethal hit by using two mechanisms. They release something called a chemical called a perforin, or perforin, which punches holes, and granzymes by exocytosis. So the perforins are going to make little pores, and then the granzymes enter in, and the granzymes stimulate apoptosis. So they basically punch holes in the cell and allow other chemicals to enter in. The cytotoxic T cells can also bind specific membrane receptors on the target cell and stimulate programmed cell death. So showing you how cytotoxic T cells, they're my favorite ones because they're like I said, little switchblade carrying ninjas that punch holes in things. There is a picture of one from an electron scanning micrograph. Natural killer cells recognize other cells that are abnormal that cytotoxic T cells might not look for, like cells that lack the class 1 MHC proteins. They also recognize antibodies that are coding target cells, and they recognize different surface markers seen on stressed cells. So natural killer cells use the same mechanism as the cytotoxic Ts for killing their target. They are also responsible for what's called immunological surveillance, where the natural killer cells and the cytotoxic T cells kind of prowl the body looking for things that they recognize as being abnormal. Then you have your regulatory T cells. Like I said, these used to be called suppressor T cells, and these are the ones that slow down or dampen the immune response by direct contact or by secreting inhibitory chemicals like interleukin 10 and transforming growth factor beta. They are important in preventing autoimmune reactions. So once your immune system has won the battle, we need to take that mass army that we made out. We don't need all of those extra soldiers in there fighting all the time. So if they continue to stay active, then they start attacking healthy tissue, and that's the basis for an autoimmune reaction. They suppress the self-reactive lymphocytes in the periphery. That's the ones that are outside of your lymphoid organs. Research into this is trying to induce tolerance in transplanted tissue. So these are the ones that they're looking at. So when people have transplants, they don't have a rejection. So just a simplified summary, as they say, of the primary immune responses, what's going on with those T cells and those B cells. So it's a complicated war that's going on for sure. And then the cells and molecules of the adaptive response. So this is a nice summary chart that allows you to put all this stuff together that I've been talking about here. So I would make flashcards. That's how I would study this. I would make flashcards for the name of the cell and then what it does. And realize there's going to be some overlap. So let's kind of switch gears for a second and talk about organ transplants and the prevention of rejection. So most common the most common type of organ transplant that happens is called an allograft, which means that we're transplanting from another human. All right. We can do xenografts, which is when we're taking tissue from another species. Not so uncommon when they use like a porcine valve, a pig valve in your heart. Um, so we can have isografts. Isografts are when you have an identical twin, if you're so fortunate to have spare parts that you're taking stuff from. But most of the time, what we're talking about here is an allograft, which is from another person. The success depends on a couple of things. One is the blood typing, which we've already talked about. There are other blood antigens, the Rh factor we have to look at, and they have to look at the MHC antigens to make sure that they are as closely matched as possible. So remember that the MHC antigens are genetic, right? This is why transplants from a relative are often more successful than transplants from a stranger because you're more likely to share MHC antigens. After surgery, when we've transplanted tissue, the patient is treated with immunosuppressive therapy, and those therapies have lots of side effects. So they have to take these drugs for the rest of their life, and the problem is that they have to suppress their immune system to keep their own immune system from attacking the foreign tissue, which means that they are susceptible to all kinds of pathogens in the environment that normally their immune system would be able to catch. When we suppress a patient's immune system, their own body can't protect against foreign agents like bacteria and viral infections. So the leading cause of death among transplant recipients is infection. In best circumstances, the rejection occurs within 10 years or after 10 years in 50% of patients. So they always say that you can hope for 10 years with transplanted tissue. Moving on to immunodeficiencies. An immunodeficiency is either congenital, meaning it's present at birth, or acquired, meaning that you somehow or another get it afterwards, that impairs function or production of immune cells or molecules. Severe combined immunodeficiency, or SCID syndrome, is a genetic defect which means that you don't have an immune system, basically. You don't have enough B cells and T cells. The defective adenosine deaminase, ADA enzyme, that allows for the accumulation of metabolites that are lethal to the C cells, and it's fatal if it's untreated. It's really, really, really uncommon, though. So the treatment for this is, of course, bone transplants. Hodgkin's disease is an acquired immunodeficiency that causes cancer of the B cells, which depresses the lymph node cells and thus leads to immunodeficiency. Acquired immune deficiency syndrome, or AIDS, is caused by a virus called the human immunodeficiency virus, or HIV, but basically takes out the helper T cells. Characterized by a severe weight loss, by night sweats, by swollen lymph glands, and then opportunistic infections take over, including cystitis pneumonia and Kaposi sarcoma. HIV is transmitted via body fluids, blood, semen, and vaginal secretions. It can enter the body through blood transfusions, contaminated needles, sexual intercourse, oral sex, and it can be passed from mother to fetus. And HIV again destroys the helper T cells, which means that you basically knock out most of your immune system. HIV multiplies in the lymph nodes throughout the asymptomatic period, which can be 10, even 15 years if untreated. During that time, you have no idea that you are infected unless you have had an HIV test. The symptoms begin when the immune system collapses. The virus can ultimately invade the brain, which leads to dementia. HIV-coated glycoprotein complexes attach to the CD4 cells. The HIV enters the cell and uses the process of reverse transcriptase to produce DNA from viral RNA. So that's just the opposite of what we know. So when we learned about transcription translation in Chapter 3 of Bio 210, what we learned was that we take DNA and we are able to copy it into RNA to make protein. And so one of the challenges of figuring out how HIV was doing it was they found out that it was actually doing it backwards. It actually goes from RNA to DNA. All right, and because it is backwards, it's prone to a lot of transcription errors, and it has a lot of mutations in it, and it's very resistant to drugs because you have a lot of mutations. So a lot of drugs are meant to target some aspect of the DNA or RNA sequence, and when it keeps changing, it no longer can work. The DNA copy of provirus directs host cell to make more viral RNA and proteins, which enables the virus to reproduce. There is no cure for AIDS. There are four major classes of antivirals that work together in a drug cocktail that help, but so far the virus has continued to become more resistant. Autoimmune diseases result when the immune system no longer recognizes cells that belong to the body. So it doesn't recognize self cells, and it thinks they're foreign and attacks them. Autoimmunity is the production of autoantibodies and synthesized cytotoxic T cells that destroy body tissue. So some examples of autoimmune disease include rheumatoid arthritis, which destroys your joints, myasthenia gravis, which impairs the nerve and muscle connections, multiple sclerosis, which is a demyelinating disease. It destroys the white matter on motor neurons, Graves' disease, which causes hyperthyroidism, type 1 diabetes, which destroys the pancreatic cells, systemic lupus, which affects multiple organs, and glomerulonephritis, which damages the kidney. How do we treat autoimmune diseases? We suppress the entire immune system. So anti-inflammatory drugs like corticosteroids help a lot. Those help to block the cytokine action and they help to block the co-stimulatory molecules. There is lots of research in autoimmune diseases looking at things like activating regulatory T cells or inducing self-tolerance using vaccines, directing antibodies against the self-reactive cells. So research continues in that area. When we have failure of self-tolerance, autoimmunity may be caused by a weakly self-reactive lymphocytes that are activated by foreign antigens that resemble self-antigens. So the antibodies against the foreign antigen may cross-react with the self-antigen. And then we have the appearance of new self-antigens that seem to be generated by mutations or changes in the self-antigens by hapten attachments or infectious damage or the release of novel self-antigens by trauma or barrier. So when we talk about why do they happen, that's what they're saying here. Why do these autoimmune diseases happen? There's a lot of different answers as to why or possibilities as to why. Hypersensitivities, this is an immune response that's overblown to something that's typically harmless. So different types that are distinguished by their time course and whether the antibodies or the T cells are involved in the process. Antibodies cause immediate and subacute hypersensitivities. T cells cause delayed hypersensitivities. So immediate hypersensitivities, also called acute or type 1 hypersensitivities, better known as allergies, begin in seconds after contact with the allergen. The antigen then causes the allergic reaction. The initial contact with the allergen is asymptomatic, so you're exposed to it one time early on and then you don't realize it, but it's the second time you become sensitized. So it's the second time or the fifth time or the 100th time that you're exposed to something that you've already been exposed to that you have the reaction. Activated IgE, it acts against the antigen. It binds to the mast cells and the basophils. Later encounter with that same allergen causes a flood of the histamine released from the IgEs, which results in the induced inflammatory response. So again, you don't have a reaction the first time you're exposed to it. It is a subsequent. Histamines cause vasodilation and leakiness of the vessels, which leads to things like runny nose and itchy eyes and watery eyes, or itchy hives and watery eyes. Asthma can occur if the allergen is inhaled, and antihistamines help to control this. Allergic reactions can be local or they can be systemic. Locally, it involves mast cells of the skin or the respiratory or the gastrointestinal system. Systemic response is called anaphylactic shock. Way more serious issue here. This is seen when you have injected allergens like bee stings, but you can also have this when you inhale things like peanuts. There's certain things that people have serious allergies to. What happens is the bronchioles constrict, which is in the lungs. It makes breathing difficult. The vasodilation results in a low blood volume, which causes a circulatory shock. And the treatment for this, of course, is an EpiPen. So they're just showing you the mechanism of an acute allergy here. So we have the antigen that invades the body. The plasma cells produce a lot of class IgE antibodies against allergen. The antibodies attach to the mast cells in the body tissue. Then you have a second response. So you have another time when you are exposed to that same allergen. So more of the antigen invades the body. The antigen combines with IgE and attaches to the mast cells, which triggers degranulation and release of the histamine and other chemicals. vessels, and the histamine causes the blood vessels to dilate and become very leaky or permeable, which causes edema. Edema stimulates a lot of mucus, causes smooth muscle to constrict or contract, and so if the respiratory system is involved, then asthma may ensue. Subacute hypersensitivities are caused by IgM and IgG transferred via blood plasma or serum. This has a slow onset of 1 to 3 hours and a long duration of 10 to 15 hours. The cytotoxic type 2 reactions occur when the antibodies bind to antigens on specific body cells. They stimulate phagocytosis and complement-mediated lysis of the cellular antigens. This is what happens when you give someone the wrong blood type. Immune complex type 3 hypersensitivity, the antigens are widely distributed in the blood or in the body. The insoluble antigen antibody complex forms. The complexes can't be cleared. So we talked about the different ways that we can clear them. In this case, it can't clear them. And so you have intense inflammation, local cell lysis, and cell killing by the neutrophils. And this is what happens in lupus. Delayed hypersensitivities are type 4. They have a slow onset from 1 to 3 days. The mechanism depends on helper T cells. The cytokine-activated macrophages and the cytotoxic T cells are what cause the damage. Some examples of this are allergic contact dermatitis like poison ivy, or if you've ever had a reaction from a lotion that you changed or a soap or anything like that. The agents act as haptens. The TB test also depends on this type of reaction. All right, that is the end, finally, of the immune chapter. I'm sorry that was such a long one. Immune system is pretty complex. This puts you in a position to study and to take your quizzes, and I will see you for the next chapter.